Right, so yes, Kelsey Moody runs Ichor Life Sciences, which, as you know from the first session yesterday, is the company in its CRO capacity that is performing the terrifying lifespan study that we're doing with 1,000 mice. But that's not how it started. And Kelsey's going to talk about a much earlier project which was spun out into Ichor from Sense Research Foundation and which has its origins in an idea I had back in 1999. So here we go.
And mic check, we good? Awesome. Alrighty, thank you, Aubrey, for the kind introduction, and everyone for coming out to discuss Tales of the Molecular Garbage Man, appropriate for this session. Before I dive in, I'm CEO and founder at Ichor Life Sciences, also our portfolio company, Lysoclear, which Aubrey's asked me to speak about today. And by way of financial disclosures, you can basically assume I'm one of those evil corporate tyrant types, and everything that I'm about to talk about today, I have some flavor of vested financial interest in. And in terms of topics that I'd like to discuss, I want to start off with the case for the eye as an organ system of choice for translating longevity therapeutics. You'll see across Ichor's entire portfolio that when possible, we have a preference for ocular disease as a stepping stone to move into systemic anti-aging drug development. In that capacity, we'll discuss a little bit of an overview of age-related macular degeneration, the specific ocular disease we're looking to target, discuss some of the foundational work performed by Methuselah and Sens Foundation, developing lipofuscin catabolizing enzymes, and then of course our current work moving this into an IND-ready state. So to start off with, I think the eye is a wonderful organ system for translating genuine anti-aging drugs, and there's a handful of reasons for that. Regardless of whether you subscribe to more of an aging hallmarks model or the 7 pillars of Sens, which I have listed here, any disease that truly is, or any process which is a fundamental aging process, its mechanism should have some flavor of ocular manifestation. When we think about moving discovery stage assets into clinical trials, no one talks about the really boring stuff like PK, for example. But if I have a drug that might work well in a systemic setting in, say, a mouse or a rat, it's a huge issue when I try to scale that up for a human. We have first pass effect for the liver, which significantly reduces the amount of bioavailable drug. In the case of your kidneys, you can just urinate out the drug depending on whether it's a large or small molecule. And these issues are a lot less prevalent when you're working in ocular disease. The vitreous itself, so the central gelatinous area inside of the eye where we deliver these drugs, also acts as a bit of a reservoir, almost time release, and there's certainly time release technologies where you can really tweak what level of drug exposure you get with whatever your either biologic or small molecule of interest might be. Additionally, when we think about some of the novel therapeutic modalities that offer some of the most immense promise in the future for age-related disease, like George Church just talked about, gene therapies, or also cell therapies, you know, we have immune issues surrounding administering those systemically that don't exist necessarily in the immune privileged space of the eye. I like eye for efficacy because how do we tell, short of running a lifespan study in humans, whether our drug is safe and effective? Well, in the eye, we have a variety of non-invasive technologies ranging from just fundus imaging through OCT to assess the safety in a non-invasive manner. And we also have very straightforward but quantifiable functional endpoints such as visual acuity. And finally, another boring topic no one talks about with drug discovery is manufacturing. So how do you make enough product on scale for systemic delivery, particularly if you have first pass effect and some of these sorts of issues? If you're trying to treat an ocular disease, you need a lot less product. So in emerging areas where the manufacturing might not be completely sorted, certain types of gene therapy and cell therapy and so on, there's an advantage in going in the direction of an eye as a starting point. And it's probably not surprising then that some of the earliest and now in clinic, what I would consider first round of anti-aging technologies are choosing the eye as a primary clinical focus. You know, Unity with their ocular program for senolytics, and then Lineage Cell Therapies with RPE derived from human embryonic stem cells. So in terms of the broad landscape of everything Ichor does, from contract research to our venture arm, which includes academic projects and portfolio companies, you know, our whole purpose in setting up our organization was to be able to systematically attack the various pillars of aging. And as our projects reach maturity and become independent spin-out companies, we have a variety of companies in our portfolio, some of them internally developed, some of those we collaborate with or fund third parties, and our goal is to comprehensively attack all of these SENS pillars. But for the purposes of today's talk, because there's a lot of stuff we can talk about that we certainly don't have time to, we're going to focus on just one of our portfolio companies, our oldest flagship, called Lysoclear, which, as Aubrey alluded to, was the technology that we've been developing was ideated by him more than a decade ago. So before we can present Lysoclear itself, I'd like to provide just a brief overview of age-related macular degeneration as a disease of interest. And you know, macular degeneration is the primary cause of vision loss in advanced age. It's characterized by the loss of the high acuity central vision. So, you know, if you're looking straight ahead and you kind of have your hand to the side, that peripheral vision isn't what's affected. It's the high acuity central vision that really allows you to engage in the world, to see people, to read, to watch TV, whatever. And if you think about the idea of losing vision, that's terrible normally, but you're talking about an aged individual who has limited mobility, limited ability to hear, losing their vision, and that can be incredibly isolating and from the patient perspective, absolutely devastating, even more so than perhaps it might be in a younger individual. When we look at kind of the anatomy of the eye to understand the disease onset and progression, this is a cross-section of the eye. I don't have pointer. Okay. If you look in the posterior eye segment, the back of the eye, this is called the retina, and this is the area where light gets focused in order to allow us to see. And you'll see just above the optic nerve a little indentation in the retina called the macula, and this is the anatomic feature that gives us that high acuity central vision. If we zoom in just a little bit more, we can appreciate there's a variety of different cellular layers as we go from inside of the eye through the posterior segment, starting with various types of neural cells that communicate information by way of the optic nerve to the brain that we interpret as vision. A little bit further back, we have our various photoreceptors, which are comprised of rods and cones, and these are the cells that get hit by light that kick off the whole phototransduction pathway. Now, as a general rule in biology, being hit by light is a bad thing. We think about sunburns in the summer as an easy example, and our photoreceptors are no different. They're constantly being hit by light, so the photoreceptors, particularly the portion of them that gets hit by light, needs to be turned over on a recurring basis. And it's the most posterior cells, the retinal pigmented epithelial cells, or the RPE, that are responsible for turning over the photoreceptors. So our photoreceptors are always growing from the nucleus towards the RPE, and the RPE cells are always gobbling them up on the other side, and this allows us to have a healthy turnover of damaged photoreceptors. In the case of age-related macular degeneration, as just a byproduct of the visual cycle, there's intracellular junk, collectively called lipofuscin, that accumulates in the lysosomes of RPE cells. This lipofuscin is chemically distinct from lipofuscin that accumulates in other areas of the body, and its predominant chemical species is a class of molecules called bisretinoids. They're vitamin A derivatives. Now, in the early stages of macular degeneration, we have lipofuscin accumulating in the cells, and the cells are able to tolerate this for quite a while. But eventually, the accumulation of this junk becomes pathological. Now, in the earliest clinically observable stages of the disease, the RPE cells can't handle the amount of junk that's accumulating, so they dump it. And in a clinical setting, we see this as extracellular junk, which we call drusen. So we can see in figure A, the little white dots in the fundus image. That's extracellular drusen that the RPE cells have deposited as a function of trying to handle this intracellular burden. If we do a cross-section of the eye and look by optical coherence tomography, or OCT, we can see a couple anatomic features. We see a little divot, which is the macula in the center, and that thick white line on the bottom of figure A, that's our RPE layer, and you can see the little bumps where the drusen has been deposited. Now, really importantly, our photoreceptor layers in this circumstance are not disrupted. The RPE cells are not dead and disrupted. So in this early stage of the disease, a clinician can see that a patient is at risk of progressing, but as a patient, I'm not observing, I'm not noticing any clinical symptoms of the disease. As the disease progresses to its more intermediate form, eventually the RPE cells can't handle the junk accumulation and they start to die, as do the photoreceptors that rely upon them. That leads to a collapse of the posterior eye segment. It presents as geographic atrophy on fundus imaging, and this starts to lead to functional deterioration of vision. Collectively, the mild and intermediate forms of the disease are called dry or atrophic macular degeneration. In the advanced stages of the disease, all of this dying photoreceptor RPE death is driving hypoxia, it's driving complement activation, all kinds of other bad things, and this can lead to new blood vessel formation, neovascularization, which can lead to bleeding into the eye and retinal detachment. This is the most severe form of macular degeneration called wet AMD. Importantly, we do have anti-VEGF monoclonals and other therapeutics for addressing the wet form of AMD, the most severe form. However, there are very few drugs, both approved and in the clinic, to treat the mild and intermediate forms. So there's a pressing clinical need to be able to address this problem. So, brief recap, I know I threw a lot of stuff at you. Lipofuscin, intracellular junk, drives the accumulation of drusen, extracellular junk, leads to a bunch of bad things that causes the disease. This is our disease hypothesis. And at Lysoclear, our goal is to eliminate that intracellular lipofuscin to prevent all of the downstream pathology. So, as mentioned previously, pioneering work in this space was pursued by first Methuselah Foundation and then SENS Research Foundation under Ably's guidance. And John Schlorendorn and later Max Pato identified a series of enzymes that were capable of breaking down the bisretinoids that accumulate in age-related macular degeneration. Sometime later, when others started working on the project, these enzymes were eventually abandoned because when they moved from cell-free assays into cell-based assays, the enzymes weren't able to efficiently target the lysosome, and that prevented the enzymes from being able to move forward into development. Now, the way that these enzymes were supposed to work, these enzymes are monosylated, which means they have mannose sugars on them, and it's known that RPE cells have a mannose receptor that delivers cargo to the lysosome. So it didn't make a whole lot of sense, and the theory was very sound. You know, if we have these RPE cells that have mannose receptor and we have monosylated enzymes, why are they not getting to the lysosome and performing work? It was around this time I had just started medical school, and with support from Life Extension Foundation, Bill Falloon and Ben Best, I had raised a large grant to start Ichor Life Sciences. And like every self-respecting medical student, I started it in my apartment. And it was with this infrastructure that I wanted to look at why the enzymes weren't able to be efficiently delivered to the RPE cells for degradation. Now, as mentioned, it's been widely published in every model system that mannose receptor is expressed both in healthy and diseased RPE. So I was really curious as to why this wasn't working in this kind of development program. Fortunately, in my living room, I was very well equipped to answer such scientific questions. When I left California to start medical school, like any self-respecting medical student, I brought a liquid nitrogen dewar with me with all the relevant cell lines I'd played with in my previous life, including ARP19, the cell type that had been used previously. I had a flow hood, I had incubators, so I was able to culture these cells, and I had a flow cytometer, which is an instrument that's used to query the expression of surface proteins. I was able to convince my very benevolent sales rep to give me some free samples of an anti-mannose receptor antibody, and I showed definitively in the living room that the ARP19 line that was used for the initial screening doesn't express mannose receptor. Well, that was really weird because we know that these cells should express mannose receptor. So I bought a vial of primary RPE from Lonza and showed, yes, in fact, the receptor's expressed. And then as I cultured that through subsequent passages, we saw a passage-dependent decrease in mannose receptor expression until about passage 4 or 5 when it was completely eliminated. And this is known, a lot of times when you take cells outside of the body, it's different. Ex vivo cell cultivation is different than if you're in vivo. And so this actually gave us a lot of confidence that this could be a viable translational path to move forward with. I'll also do a selfless plug. If any of you are grad students or in med school and you want to know how to set up a living room laboratory, we did bother to submit and we're peer-reviewed publication on how to do that. So that's something that exists out there if anyone cares. So over the couple years following that, we did a ton of work with manganese peroxidase, a recombinant manganese peroxidase, as our therapeutic lead. I mentioned that lipofuscin is comprised of a variety of different bisretinoids, so we synthesized every one that was able to be made and showed that manganese peroxidase has very efficient degradation of all of them. That's the graph on the left. Importantly, you'll note that the lipofuscin fluorophore that is the most resistant to degradation is A2E. So we chose to use that as a primary readout both for cell and in vivo studies on a move forward basis. The rationale being whatever we saw with A2E, the actual effect is probably better, but we wanted to have the most pessimistic readouts possible. In the center, we took RPE19 cells and loaded them with A2E, and we saw that untreated, they had a 90–95% viability. A2E cytotoxic and killed the cells. By forcing manganese peroxidase into the cells, we were able to rescue some of the A2E-mediated damage, and manganese peroxidase by itself was non-toxic. We then moved into an ABCA4 mouse model. This is a murine model that has accelerated A2E accumulation, and we showed that administration of this enzyme allows us to break down A2E in vivo, and this was the first demonstration of in vivo efficacy for an enzyme therapy for macular degeneration. But we had a problem. We were very excited to move into the clinic with this, but our targeting system created a bit of an issue. So manganese peroxidase, the way it gets into the cells, as we discussed, relies upon mannose sugar chains. And the way that these chains grow is they're kind of like a tree in that the branches kind of grow somewhat at random. And that degree of heterogeneity prevented our manganese peroxidase, despite very favorable safety and efficacy profile, from moving in as a viable clinical candidate. Of course, all of this is going on, and we're not static. So, where we were when we started this in 2014, 2015 is very different from where Ichor is now. And over the years that we were working on this project, we've built Ichor out into a 60-person organization with full preclinical drug discovery capabilities, comprising 3 facilities, all based in New York State, both in Potsdam and Lafayette, New York. So we were equipped to take on a more advanced program to iterate through what was discovered with manganese peroxidase. We did a lot of mechanistic work to identify specifically how the manganese peroxidase was capable of breaking down A2E and other bisretinoids. I'll skip over this in the interest of time now. Suffice to say that we believe the mechanism is through a heme-mediated oxidation. And with these insights, we set criteria to go searching for new enzymes. Our selection criteria was fairly simple. We wanted targets that had sufficient redox potential to break down A2E based on our mechanistic insights. We didn't want to have the sort of manufacturing hurdles that we had encountered with manganese peroxidase, which has to be expressed in Pichia pastoris. It's glycosylated with a lot of disulfide bonding. So we wanted to have a product that could be expressed readily in bacterial culture. And we wanted the source species to have optimal growth at acidic conditions, so a pH of 5, because the lysosome is where we need these enzymes to work. And armed with this information, we did a massive literature review, massive screen of all kinds of different potential candidates that we could move into development. And from that, we selected 28 unique enzymes from very diverse species that were included in our screening cascade. We did expression testing, which we started with pilot expression in shakers, scaled up to bioreactors for gross yield determinations. We then went into purification with a purity assessment. We're concerned about CMC issues, such as homogeneity of the enzymes. Are they aggregation prone? What's the relative bioactivity? And with that information, we were able to identify pure yield determination. Importantly, when you're dealing with biologics, particularly enzymes, aggregation proneness is a big issue that we want to pay attention to. And several of the enzymes we developed actually had surface cysteines that weren't part of their actual function or structure, but they did lead to these enzymes being aggregation prone. So we also did a mutational screen to eliminate those cysteines to make a more viable lead therapeutic. We then moved our best leads into cytotoxicity and cell-based efficacy studies, and then mouse, rat, and rabbit studies. And we were very encouraged to see every single one of the enzymes that we were able to express recombinantly was able to efficiently degrade the lipofuscin fluorophores. I have representative data just for 3 of those. As with the previous figures, the graph on the left is showing that the enzymes are capable of breaking down lipofuscin bisretinoids in cell-free systems. Importantly, if you look at the, so leftmost is BSA, our negative control. Our manganese peroxidase is the second set of columns. But if you note, manganese peroxidase does not have any specificity towards an acidic pH. Because acidic function was important in our screening, we see that all of our enzymes that we selected actually have preferential activity. They degrade more of the fluorophores at acidic pH than at physiologic. We moved those enzymes into cell-based screening in the center, and they performed well, and then ultimately in vivo studies, which is shown on the right, again using our ABCA4 model. So collectively, we think we have a whole panel of enzymes that are effective for degrading these fluorophores. What was really interesting is because of the breadth of species screened, we saw very, very low primary sequence homology among these hits. So the genes, the DNA that encoded for all of these enzymes were completely different for each of them. However, when we looked at the tertiary structure, we see that the structures of these proteins are actually highly conserved. And this is exciting to us because this built out a very strong IP position, gave us a very large quantity of viable leads that we could move forward with, and most importantly, this is all mechanistically informed. So all of our proof of concept studies delivered these enzymes just using cell-penetrating peptides. And so now that we think we have really good enzymes that can move forward into development, we need to select the best vehicle for delivery. And what we settled on was lipid nanoparticles. So we have a collaborator who specializes in the development of lipid nanoparticle technologies and their translation. And we've screened, this is just the first 10 nanoparticles or so that we've screened. So they gave us a variety of nanoparticles loaded with GFP. Pretty much all of them worked at various levels. You can see group 2 there worked particularly well. And so we've gone through and done a massive screen of a bunch of different lipid nanoparticles to identify which ones are going to to deliver our therapeutic payload most efficiently to the RPE cells of the eye. As of last week, our lipid nanoparticle screening campaign is complete, and rather than use the nanoparticles to deliver the enzyme, which will have limited therapeutic payload, we're delivering mRNA so we can have very high relative expression of these enzymes. So we're optimizing lysosomal targeting using these mRNA expression constructs, and our intention is to start pivotal in vivo studies by fall of this year, with the eventual goal of a drug candidate for IND-enabling studies by spring of 2024. Of course, all of this is just one project within one company, within one division of kind of the Ichor ecosystem. If there's other areas in the aging space that are open for collaboration or things you'd like to talk about, this is just a small piece, of course, of what we do at the company. We have received National Eye Institute funding and, of course, investor funding for this project, names of some of our team that were pivotal for this. And with that, I thank you for your time, and we'll take any questions.
Well, great job, Kelsey. I must say, I'm very proud of a lot of my people who I could call my protégés over the years, but Kelsey's right up there among them. Actually, I'll start with a question of my own. And it arises from the last thing that you mentioned, the fact that you have transitioned the delivery from delivering enzyme to delivering mRNA. So what that means, of course, is that your enzyme is not synthesized in bacteria anymore, it's synthesized in the mammalian cell. Have you encountered any advantages or disadvantages arising from that, like post-translational modifications that are different in the two circumstances?
No, that's an excellent question. We haven't encountered any issues with post-translational modifications of our top enzymes as we've tried to transition them to mammalian expression. We actually think having a simpler system, like a microbial the expression being optimized for microbial expression means when we start playing around with lysosomal targeting motifs for the mRNA delivery, we think that's going to be a lot simpler, and we're going to have a lot less translational difficulty because these are structurally simpler enzymes that have no post-translational modifications and are generally simpler than their, say, fungal counterparts.
Mm-hmm. Yeah, makes sense. All right, any questions? Over there, Tillman has a question. I'm not surprised.
Thank you. Great work. Actually, I have two short questions. Glycosylation is changing during aging. Do you have different efficiency in delivering and during different age groups? And the second one, in RPE cells, if they are loaded with lipofuscin, what about the pH? Is it not buffered as in other cells?
Both excellent questions. So I don't have a strong opinion on the difference in glycosylation states from young to old. Because we're not relying upon mannose delivery or glycosylation, we've kind of sidestep that as a development hurdle for us, but certainly for other therapeutics, that's something to keep in mind and something that could be very relevant. In the case of lysosomal deacidification, which is certainly of concern in the RPE, because as this lipofuscin accumulates, the pH of the lysosome changes. There is a balance to be struck there. All of these enzymes that we're interested in potentially advancing do have activity at higher pHs as you approach physiologic. So they're not only active, it's not binary. They're not only active at acidic pH. What we don't want to have, if there's any leakage in the expression, we want to avoid to the extent possible having enzymes that are optimally active at physiologic pH as a safety concern. But with the enzymes that we have, we haven't observed that to be an issue, and they seem to be performing admirably despite the lysosomal deacidification.
One more over here in the middle.
Hi, thank you so much. I think your work is amazing. I just want to ask you, when the enzymes break down this lipofuscin, is there no byproduct still around in the cell that could be harmful?
Yeah, that's an excellent question. We have not fully characterized the breakdown products. in part, that's really difficult to do because if you think about lipofuscin, it's not a single thing, right? It's comprised of a dozen different bisretinoid fluorophores, plus all of the other junk that those fluorophores drive the accumulation of ceramides, different sphingolipids, lipids, et cetera. So being able to parse apart all of the different breakdown products for all of that isn't really feasible at this time. We are certainly paying close attention to cytotoxicity. What I can say is if we load up cells with A2E or other bisretinoids, hit them with the enzymes to degrade it, we don't see any cytotoxicity as a result of that. But that's certainly something we're going to be paying attention to. We have done some work characterizing the mechanism by which the primary fluorophore A2E is broken down. Our current thinking is that the breakdown products are probably things that can be catabolized normally within the lysosome, and I've got all kinds of mechanistic stuff we could talk about afterwards regarding that topic, but that's our current thinking on the issue.
Thank you so much.
Great. I think it's time for the next speaker. Thanks
All
very
right,
much.
thank you,
Kelsey.
Aubrey.